Okay, so hi everyone, my name is Farhana. I'm a PhD candidate at the University of Toronto. Thank you for letting me share my research with you today. I will be talking about personalized medicine for the treatment of depression. So before I jump into the topic, I want to give a little background about myself and why I was invited by your lovely teacher, Arno, to speak about this topic. I graduated from the mass program at Danforth. I was seated exactly where you are now many, many moons ago. Then I went on to do a honoris Bachelor of Science degree in biochemistry and neuroscience at the University of Toronto. During my undergraduate studies, I worked on 2 research projects and also volunteered at a lab. And from these experiences, I realized that I really enjoyed research in neuroscience. I enjoyed the process of designing experiments, gathering and analyzing data, making sense of the data, and sharing the results with the larger research community in the form of a paper or a poster. As a result, I went on to do a master's degree in neuroscience. I further developed my research skills and interests. I then decided that I can possibly see myself doing a career in research. So I went on to do my PhD, which is a lot of studying. Currently, I'm a PhD candidate at the Department of Pharmacology and Toxicology at the University of Toronto. I conduct my research in the Pharmacogenetics clinic at the Center for Addiction and Mental Health under the supervision of Dr. Daniel Mueller. So as you may know, that CAMH is a psychiatric hospital that is located downtown to the corner of Spadina and College. The focus of the research our lab does is exploring the effects of genetic variation on response to psychotropic medications. And what these are medications that affect the brain, such as antipsychotics and antidepressants. The focus of my doctoral research investigates the genetics of antidepressant response for the treatment of depression, which is what I'll be talking about today. First and foremost, what is depression? Depression is a common term we have often heard talked about in relation to mental health. According to the American Psychiatric Association, depression is a mood disorder that is associated with feelings of sadness and or loss of interest in activities you once enjoyed. It can lead to a variety of emotional and physical problems and can decrease your ability to function at school, work, and at home. Depression is a common illness worldwide, affecting a total of 280 million people. It's a lot of people. About 5% of adults older than 18 are affected, and about 2.8% of adolescents 15 to 19 years old are affected. Although depression affects so many people, we do not know, or we do not fully understand that the biological processes that cause depression. And as a result, the treatment of depression remains a huge challenge. One approach to treating depression is with medications called antidepressants. With antidepressants, however, it's not a foolproof method of treating depression because depression is not like cancer or like a heart disease that you can change your lifestyle or take a medication to improve your symptoms. Depression is very complex. So within people who take antidepressant treatment, about 2 in 3 patients continue to experience depressive symptoms following their first course of antidepressants, and 10 to 20% of patients do not respond at all despite multiple drug therapies. Currently, antidepressants are prescribed by physicians using the trial and error approach. So how this works is that the physician, usually a psychiatrist, diagnoses a patient with depression based on a set criteria and deems that antidepressant is the best course of action for treatment for the patient. The physician prescribes the same medication and the same dose to all the patients that come in to the clinic with depression. With this trial and error approach, there are large differences between individuals in terms of how they respond to medication. So there are some who will benefit from the medication, some who do not benefit at all, and some who actually show an adverse reaction to the medication. This is not the most ideal method to prescribe medication because not everyone will respond, and only a small segment of patients will benefit from treatment. So why do patients respond so differently to their medication that some patients respond, some show no response, and some show adverse response? This variability in medication outcome is caused by several factors, including age, gender, ancestry, clinical characterization, such as other diseases the person may have, drug-drug interactions, such as other medications that the person may be taking, as well as the antidepressants, and drug-gene interactions. This talk will focus mostly on drug-gene interactions. So this slide is a very brief review of material you probably already know. So humans have 46 chromosomes, and each cell of their body, excluding the gonads, and 23 of the chromosomes come from the mom and 23 from the dad. And in this example, this is a chromosome pair. The dark blue is from the father and the light blue is from the mother. And on each chromosome are segments of DNA called genes, and it's depicted as bands on the chromosome in this image. So that this gene here shown encodes hair color, for example, and one copy of the gene is from the mother and one copy is from the father. So genes are what contains the blueprint for our body. All human beings are 99.9% identical in their genetic makeup, which is why we have the same eyes, the same like structures of our body. Like our eyes are located at the same place, our lips are located in the same place. So that's because we are 99.9% identical. And the remaining 0.1% is what makes us different from one another. And this 0.1% difference in our genetic makeup contributes to why we respond differently to medications. So personalized medicine takes into account an individual's unique genetic makeup to identify the best medication and dosage for the patient. This approach sounds great relative to the trial and error approach, which we see here, because patients can be prescribed medication based on their genetic makeup, which optimizes their response to the medication and decreases their chances of side effects. So each patient benefits from personalized drug choice. However, there's one major issue with this approach. We currently don't have genetic markers that reliably predict response and side effects to antidepressants. There isn't one single gene for antidepressant response and side effects. So we cannot really just choose a gene, just select a gene and determine what medication a person will be prescribed based on that one gene. So this is where pharmacogenomic research comes in. The goal of research in this field is to identify genes that predict response and side effects to antidepressants. And once these genes are identified, these genes can be used in a pharmacogenomic testing panel to help physicians choose the right medication and the right dosage for the patient to optimize their treatment benefits. So here we see patients receiving different medications and different dosages based on their genetic makeup. And the end goal is we will have patients who will benefit from personalized drug choices. So you may recall the central dogma of biology states that segments of DNA called genes are transcribed into messenger RNA, which is then translated into polypeptides. These polypeptides fold on top of each other in a particular configuration to form proteins. Therefore, differences in our genes can lead to differences in the function of proteins. So to identify genes related to antidepressant response and side effects, we can take a look at the proteins that the antidepressant interacts with when it's in the body, such as enzymes like liver enzymes. So when it's ingested, it interacts with liver enzymes to be metabolized, and it interacts with transporters like the serotonin transporter, and it also interacts with receptors like the serotonin receptor, so it binds to the receptor and causes release of serotonin. So these are all different proteins that the antidepressant interacts with, and these proteins are encoded by DNA, which may or may not vary between individuals. So the remaining portion of this talk will focus on liver enzymes that process antidepressants. These liver enzymes are called cytochrome P450 enzymes, which I'll refer to as CYP enzymes. So when the antidepressant medication is ingested orally, it is absorbed through the intestinal epithelium and passed to the liver. And in the liver, the antidepressant is processed and released into the bloodstream and then carried to the brain where it affects neurotransmission. And that's how you see an individual improving in terms of their depressive symptoms. As you can see, there are many different CYP enzymes in the liver, but not all of them are involved in processing antidepressants. Majority of antidepressants are processed by CYP2C19 and CYP2D6, and these two enzymes have been very well studied in pharmacogenetic research. So there is genetic variation in the genes that encode CYP2D6 and CYP2C19, and it's associated with altered enzyme activity. So as a result, individuals can be categorized into one of four metabolizer phenotypes based on their genotype. So the first metabolizer phenotype are ultra-rapid metabolizers, and these individuals have multiple copies of the normal gene, resulting in an increased rate of metabolism. So they're processing antidepressants at a faster rate than a normal metabolizer would. Second, we have the normal metabolizers, and they have functional copies of the gene and therefore have normal drug metabolism. So they have two copies that are normal, and therefore they have a normal rate of metabolizing the drug. So the drug is in their system a good amount of time to have a therapeutic effect. And we call this group the wild type group. And then we have the intermediate metabolizers who have one functional and one non-functional allele, one non-functional copy of the gene shown in red, and they have a decreased rate of drug metabolism, which means that there is higher levels of unmetabolized drug in their blood. So a lot of the drug is not being metabolized. And then we have poor metabolizers, which have two defective copies of the gene. And that results in most of the drug being unmetabolized. So most of the drug that is ingested is unmetabolized and does not go through processing. For CYP2C19 in particular, there is a fifth additional metabolizer status. I just wanted to mention this on the side. And these are rapid metabolizers who have one normal function and one increased function copy of the gene, which means they are somewhere in between ultra-rapid metabolizers and normal metabolizers. So categorizing genetic variation into phenotypic groups helps to distinguish the enzyme's level of activity in each individual. For instance, for someone who's a poor metabolizer, most of the medication that they ingest is going to be unprocessed, and that will affect how much of the medication passes through the blood-brain barrier into the brain and how it affects neurotransmission. So these people may show decreased response because they have more of the drug that is not processed by the liver. So this slide is an example of a pharmacogenetic study that I've conducted. In this particular study, patients with depression are given an antidepressant called escitalopram, which I'll refer to as ESC. And ESC is metabolized by CYP2C19 and CYP2D6 in the liver. And what we did is we looked at how much each patient improved in their symptoms based on their metabolizer phenotype, based on whether or not they're a normal metabolizer, intermediate or poor metabolizer, or a rapid or ultra-rapid metabolizer. So as you can see, the normals are in blue. The intermediate and poor metabolizers are in yellow, and the rapid and ultra-rapid are in pink. So this is an example of how a result graph looks like in a research paper. And we have in the y-axis how much they improved from when they first started the drug. And we have on the x-axis the week. So from week 2 to week 16, we see that every patient improved, but we see that the ones who are intermediate and poor metabolizers, they improved the least. So because they're processing the medication very slowly, they're not showing improvements as much as the normal metabolizers or the rapid and ultra-rapid metabolizers. And this one is for CYP2C19. When we look at CYP2D6, we have the normal metabolizers in comparison to the intermediate and poor metabolizers, and we don't really see a difference in terms of how much they improve relative to each other. So what we can say is that treatment response is different between metabolizer phenotypes for CYP2C19 only, but not CYP2D6. So for those who have slower enzyme metabolism, have lower levels of response over time compared to those who have normally functioning enzymes for CYP2C19. Whereas for CYP2D6, it doesn't really matter if they're normal or slow metabolizers, they improve at the same rate. So what do these findings mean? So it shows that there is strong evidence that genes that encode liver enzymes CYP2C19 contribute to citalopram response. CYP2C19 and CYP2D6 can be used in a pharmacogenetic testing panel to help physicians choose the right medication and dosage for patients with depression. Sorry, this should only say CYP2C19, not CYP2D6, because CYP2D6 did not show an association. So CYP2C19 can be used in a pharmacogenetic testing panel to help physicians choose the right medication and dosage for a patient with depression. For instance, since CYP2C19 intermediate and poor metabolizers respond less to citalopram compared to normal metabolizers, they may be given a lower dose of the medication, or the physician may select a different medication altogether so that they respond better to treatment. So I know there's a lot of information to process, especially through a whole study in there. So I'll just summarize in terms of what the take-home message is. The take-home message is that treating depression is challenging because there is a large variability between individuals in how well they respond. Genetic factors contribute to this variability in response to antidepressants. And research is investigating variations in genes that contribute to variations in antidepressant response and side effects. And this research, the example that I gave you is a pharmacogenetic research study that looks at CYP2C19 and CYP2D6 genes in particular in relation to treatment response. And that's the kind of studies that is conducted. So once identified, these genes can be included in a pharmacogenetic testing panel to help physicians select the right medication and dosage for their patients in order to optimize improvements of depressive symptoms. And that is all I have. And if you have any questions about any part of this talk, or if you have questions about grad school or just undergrad, feel free to get in touch with me at this email address. Thank you for listening.